My name is Sahibjeet, and I'm excited to have this new session of Understanding Mutations and Clinical Trials Update, part of our Let's Get the Gist of It series. So before we dive into our presentation, I do want to share a few things. The Raft group provides a fair and balanced approach to supporting patients, and we do not promote any commercial or investigational products due to any financial hardships or relationships. We are not medical professionals, and we do not intend to provide any medical diagnosis or make any specific treatment recommendations. Instead, we would only provide information for you to discuss with your doctor to review. This session will be recorded and shared afterwards, and I encourage everyone to utilize the Q&A feature in Zoom to ask questions. We will have time at the end to answer those questions. Today, we will have a quick gist intro, and then we will compare the differences between a pathology report and a mutation report. Once we get a good understanding of that, we will then dive into gist mutations, followed by clinical trials update, and end the session with a Q&A. So gist diagnosis and treatment involve a multi-step process. However, the exact timeline can vary based on the individual's symptoms, general health, and specific characteristics of the tumor. On the screen is a generalized overview of what a gist timeline may look like. So the first portion is the symptom recognition and initial consultation. Whenever we speak to any GIST patient, whether that be during the time they are joining our GIST patient registry or just in general, we always ask the question, what events led up to the diagnosis of your GIST? And this is super important because everyone is unique to their event, but commonly the symptoms shared include abdominal pain, feeling full quickly, nausea, vomiting, blood in stools, anemia. And typically when these symptoms are felt, the patient goes to the emergency room to get more information on what is causing these symptoms. The next portion in the timeline is the diagnostic tests. And this is a very important part because if the doctor suspects a mass, they may order a series of tests such as an endoscopy, a CT scan, MRI, or PET scan to figure out what is causing the symptoms. At this point in the timeline, there is no diagnosis or confirmation of GIST. If the imaging shows a tumor or mass, then a biopsy would be needed to confirm GIST. In some cases, the biopsy might be taken during the endoscopy or through a fine needle inserted through the skin. The third portion, which is the tumor analysis and staging, is when once the tumor is obtained, the tumor will then be analyzed to determine what kind of cancer it is and the mitotic rate. So at this point, this is where a GIST would be confirmed and diagnosed. At this point as well, with the mitotic rate, it also lets us know the staging of the disease. At this point also, a medical professional may also order a mutation test to identify the specific mutation. Although mutational testing is not always done at this point, but through increased awareness and sharing the importance of mutational testing, it is getting completed earlier in the patient's GIST journey than ever before in the past. Treatment plan and development is the next point in the timeline. So based on the diagnosis, the medical team will provide a treatment plan. And the treatment typically starts with surgery, if possible, followed by a targeted therapy with drugs like imatinib. Some patients may start medication before surgery in a neoadjuvant setting to try to shrink the tumor and then have surgery followed by adjuvant medication. Some patients may opt out of surgery, and some patients may not need to take any medication. These decisions on treatment will depend on the size, location, stage of the tumor, and of course, the patient's overall health. The last portion in the timeline is the treatment and follow-up. So once the patient undergoes surgery and suggested treatment, the patient will also begin regular follow-ups and imaging to monitor any signs of recurrence. It's important to note that these are estimated times in the timeline provided and can vary greatly depending on the individual's patient's circumstances. The patient should have regular communication with their healthcare provider to understand the timeline and what to expect at each step. So in the previous slide, we spoke about the timeline, and one of the most important, if not the most important point in a GIST timeline is the tumor analysis and staging. Those items will be found in the pathology report and mutation report. So what's the difference and why do we need both reports? The pathology report is what is reported first. It is the report that confirms that a patient has GIST. Without a pathology report, we will not know all the key points that tell us that it is a GIST. Pathology reports are created either through a biopsy sample that may have been obtained during an endoscopy or a fine needle biopsy, or through surgery. On the report, a patient will see immunohistochemistry stainings, or IHC for short. IHC stains is a diagnostic test that tells us what kind of cancer the sample being tested is. A pathologist will apply a stain to the sample, and if the sample stains, then it is indicative that the cancer-identifying protein is present. For GIST, the most common stains to diagnose GIST are CD117 or c-KIT, DOG1, and CD34. CD117 and c-KIT are interchangeable terms and are not to be confused with KIT mutation, which is found on the mutation report. If you see CD117 or c-KIT on your pathology report, followed by a positive or a negative, then it is referring to the stainings confirming GIST diagnosis, not the mutation. The pathology report also shares the tumor measurements, the mitotic rate, which lets us know the risk and staging, as well as macroscopic and microscopic descriptions. So macroscopic descriptions include the size, the shape, color, consistency of it, and then the microscopic description shows us what they see under a microscope. So the cell shape, cell morphology, the arrangement of the cells, those items we will see from those descriptions. Now for a mutation report, this is the results from mutational testing, which is also known as genomic sequencing or molecular testing or biomarker testing. Those terms are all indicative of testing for a mutation. This is like step 2 in this point of a patient's timeline. So the first part is, or first step is to get a pathology report diagnosing a patient and confirming that it is GIST. And step 2 is figuring out, all right, now that we know GIST, what is causing that GIST? Like what is driving the GIST? This report provides what mutation or mutations are detected if a patient has a secondary mutation. It also tells us the exact location in the gene where the mutation has occurred in the amino acid. It can also provide a list of treatments that the mutation will respond to, along with any clinical trials that a patient may benefit or be eligible for. And the report also tells us the mutation frequency. So what is the mutation frequency? If a patient's cancer has been sequenced over time, the report might show changes in the frequency of certain mutations. This can provide information on how the tumor is responding to treatment or evolving over time. IHC tests for expression of protein by tumor cells. It tells us if the tumor is a GIST and it requires a tumor sample from either a biopsy or a surgery, whereas a mutational testing tests for mutations in the gene, and it tells us what mutation is driving the GIST, and it requires a tumor sample or a blood sample through ctDNA, which is circulating tumor DNA. It is important to note that patients who get ctDNA typically have a better report or a result from that blood sample draw if they have more of a tumor burden, because the way that they test the blood sample is from the tumor cells shedding into the bloodstream. So if a patient has, let's say, a small tumor or just one tumor in their body, they may not have as much tumor shed into their blood system as a patient who may have advanced or metastatic GIST, and they have more of a tumor burden that is shedding into the bloodstream. So once they draw that blood, they go ahead and circulate it and remove or extract the DNA from it, and then perform the analysis to see what mutation is driving the cancer. So now that we know the difference between pathology report and the mutation report, why is it important for us to also have mutational testing done? Mutations play a significant role in the development and treatment of GIST. Most GISTs are caused by mutations in the KIT or PTGFRA gene, which are genes that provide instructions to proteins involved in signaling pathways that control important cellular processes. In healthy cells, these proteins and genes can regulate how cells grow, divide, and survive. When mutations occur, these genes can lead to uncontrolled cell growth, leading to the formation of tumors. Understanding these mutations is crucial for several reasons. One is targeted therapy. Certain medications known as targeted therapies work by blocking the activity of the proteins produced by the mutated gene. For example, the drug imatinib or Gleevec is effective against GIST with certain mutations. Treatment response is so the type of mutation can predict how well a patient responds to these targeted therapies. Patients with some types of KIT mutations respond very well to imatinib, while those with certain PDGFRA mutations may not respond to it at all. Another reason why it's important to know your mutation is prognosis. The presence, type, and location of the mutation can also provide important prognostic information. And lastly, new therapies. Understanding the specific mutation that drives GIST growth can guide research into new treatments. For example, newer drugs have been developed to treat GIST patients with a specific PDGFRA mutation that don't respond to imatinib. Therefore, genetic testing to identify mutations is an important part of diagnosing and treating GISTs. The information gained from these tests help doctors personalize treatment plans to each patient's unique genetic profile, thereby improving the chances of a successful outcome. So today we're going to be talking about the different types of GIST mutations. We will talk about KIT, PDGFRA, SDH Deficient complex, NF1, BRAF, and Ntrack Fusion. I do want to note that Ntrack Fusion is not a mutation, but rather a fusion, but I'll discuss the difference between a mutation versus a fusion later on. We will begin with KIT mutation. So KIT mutations are the most common mutations in GIST patients. Within KIT, exon 11 is the most common, with around 60 to 70% of GIST patients harboring that mutation. KIT exon 9 is not as common, but we do see many patients with that mutation. And it is important to note that for exon 9 mutations, a higher dose of imatinib is more responsive. KIT exons 13 and 17 are most common secondary mutations we see in GIST patients. As you can see, KIT exon 13 and 17 are both imatinib resistant, and exon 13 is sunitinib sensitive, while exon 17 is regorafenib sensitive. It again shows how important mutational testing is, because mutations will tell us which treatment to be on for the optimal treatment. KIT exon 14 and KIT exon 18 are less frequent compared to 13 and 17, and those two are also both resistant to imatinib. KIT exon 14 does have studies that show sensitivity to sunitinib and regorafenib, whereas KIT exon 18 is regorafenib sensitive. Next, we talk about PDGFRA mutations, and they are the second most common gene in where mutations arise for GIST patients. PDGFRA exon 18 D842V is the most common, which is sensitive to AYVAKIT or avapritinib. When AYVAKIT was approved back in 2020, it was huge news for D842V members because there was no other drug that was responsive to that mutation. Other mutations within the PDGFRA gene, like exon 12, and non-D842V exon 18 mutations do show sensitivity to imatinib, again highlighting the importance of mutational testing. PDGFRA exon 14 is rare, with less than 0.1% with that mutation, and it is shown to be sunitinib sensitive. Here is a great image that shows TKI sensitivity based on amino acid in both KIT and PDGFRA mutations. So here we can see those that are starred red show that they are not sensitive, so they're resistant, whereas the ones that are green, they show sensitivity. Next, I want to talk about SDH deficient. So we typically term them as an SDH complex mutation, and here SDHA often shows a negative immunohistochemistry expression. This is indicative of an SDH mutation. So if a pathologist goes ahead and tests for the SDHA or SDHB immunohistochemistry stain, it can mean that that patient may be SDH deficient. It doesn't confirm it, but it is one step closer to getting a confirmation of that mutation. SDHA mutations are more commonly present in women. For SDHB, this also is indicative through IHC expression, but to be confirmed, you do have to get mutational testing to know exactly where in the SDHB mutation or gene is the mutation. Within SDHB, there is carnase dyad, which is both a GIST tumor plus a paraganglioma. For SDHC and SDHC epimutant, this SDHC is often associated with familial GIST. And then this can also cause carnase triad, which is a non-hereditary causing GIST, which is GIST as well as paraganglioma and a pulmonary chondroma. SDHC is the last of the SDH complex, and this is least commonly associated with GIST, and it's more common with hereditary paraganglioma or pheochromocytoma. Some common characteristics in SDH deficient patients are that it commonly occurs in younger patients. They can be multifocal and typically in the stomach origin. They are indolently behaved, so some patients just have surgery to remove the tumor followed by imaging for surveillance. They are imatinib and other standard TKI resistant, and genetic counseling is recommended to see if it is germline or not. NF1 mutation is something that we do see in some patients. Some characteristics for this mutation are that we have noticed it commonly comes from the small intestine, and it's usually multifocal. Similar to SDH complex, any patient with an NF1 GIST mutation, we do recommend genetic counseling. There is limited imatinib response. However, we do have a couple of patients in the patient registry who take selumetinib for their NF1 mutation. Next is BRAF mutation, and this is also more of a rare mutation that we see in GIST patients. The mutation of BRAF V600E is the most common within GIST patients. Again, given how rare it is, we see less than 5% patients, and there is a poor response to standard GIST TKIs. However, other drugs called vemurafenib and dabrafenib are sensitive and studies have shown sensitivity for the BRAF mutation. So as I had earlier said, Ntrack Fusion. So Ntrack Fusion in GIST results in the merging of an Ntrack gene with another unrelated gene, leading to a production of a fusion protein with continuous activity that drives cancer development. It is important to note that a fusion is not a mutation rather than two genes fusing together. For Ntrack, there is poor response to standard GIST TKIs, but do show promise with larotrectinib and entrectinib drugs. The Raft group is currently offering free next-generation sequencing to patients. Next-generation sequencing tests for over 600 genes at once compared to the standard kit or PDGFRA test, which only tests for mutations in those two genes. An eligibility criteria includes that a patient must reside in the United States, have not had any type of previous mutational testing, but there is, if a patient has had just the basic KIT or PDGFRA mutation test and came up as a wild type, they are eligible. Patients must have a treating oncologist, and a patient must have a tumor sample available. If you haven't had mutational testing, and I hope that this presentation so far has convinced you and given you, motivation to get mutational testing, please reach out to us and we will happily be able to provide additional details on how to get next generation sequencing done. So now we will transition over to updates in GIST clinical trials. Before we share updates, I do want to explain the importance of clinical trials. A clinical trial is a type of research study that aims to explore whether a medical treatment is safe and effective for humans. Clinical trials aid in the development and testing of new treatments. They offer opportunity of gaining access to new treatments after other therapies may have failed. Receiving treatment and care at a major GIST institution is available to those who do participate in the clinical trials, and those who participate in them are helping future GIST patients by contributing to medical research. The Raft group does have an extensive GIST clinical trials database that is managed and updated by one of our board members and allergy science member, Jim Hughes. He puts a lot of effort in keeping this up to date with all trials available for GIST patients. The website is www.gisttrials.org, and I encourage everyone to check it out. So I want to share one of the most exciting updates in the clinical trial world for GIST patients. Deciphera, who we know is the company that created Quinlock, has unveiled their new trial set to start late summer of 2023. This trial is a global trial, which is very exciting. This trial is a phase 3 open label randomized trial understanding ripretinib in a second-line setting. So the eligibility criteria is that a patient must have a KIT exon 11 primary with a secondary mutation in KIT exon 17 or KIT exon 18. They anticipate 54 participants to be enrolled. The study is a 2-to-1 randomized, where patients will either receive ripretinib 150 milligrams daily continuously, or sunitinib 50 milligrams once daily, but on a cycle, so 4 weeks on and 2 weeks off. Patients will receive the study drug until disease progression, and patients who are on the sunitinib arm may cross over to receive ripretinib upon disease progression. Patients will receive ctDNA to determine eligibility at the start of the trial. And the trial sites are not yet shared, but we will definitely keep you all updated once they share them. If you are interested in learning more about this trial, please contact Denise Evans. I have shared her email on the screen, but it is devans@liferaftgroup.org, and she will be able to provide more information and connecting you with the trial. Next, I want to share updates from the Theseus trial. THE 630 trial has unfortunately been terminated as of earlier this month. So why did they discontinue development? Theseus was attempting to understand dosing through its cohorts with a projected target exposure of 40 milligrams. Unfortunately, they had to stop due to adverse events that occurred in patients. So what were the results from the adverse events? So the reason for stopping was because 2 patients in cohort 1 experienced grade 2 or higher hypertension. 1 patient in cohort 2, so a patient who was taking 4 milligrams, experienced grade 5 myocardial infarction. 6 patients among all cohorts experienced hand-foot skin reaction, and 23 patients among all cohorts, which was more than 50%, 65.2%, experienced any grade side effects. It is important to note that out of the 23 patients, 21.7% experienced grade 3 or higher. On the left, you'll notice a table with different cohorts, dosing, and how many patients were enrolled in each. As you can see, cohort 8, they never were able to get to, as that was the projected dose target, but they had to stop while they were in cohort 7. Of course, the question of what happens to the patients enrolled who were responding great to the drug and what's next with Theseus comes up. So patients currently enrolled in the trial will continue to receive the drug until a treatment discontinuation criteria is met. So if a patient has disease progression or other adverse events, that's when they will stop. Some exciting news is that Theseus has announced a new program targeting KIT for patients with early line GIST, which is coming early 2024. So hopefully within the next few months, we will have more information and more updates for that. Cogent Bioscience's PEAK trial of bezuclastinib and sunitinib is showing great results. Earlier this year, part 1 of the trial's phase 3 was completed, and it showed encouraging safety, tolerability, and clinical activity. Side effects were minimal, with grading between 1 to 2. the combination of bezuclastinib and sunitinib did not show to add to the frequency of severity of side effects compared to just sunitinib usage alone, which is pretty awesome. The results from part 1 are still too new to provide progression-free survival data. The trial is now enrolling patients in part 2, and this portion is open to patients who have progressed on or are intolerant to imatinib only. So again, second line. Participants will be started on the selected dosage of 600 milligrams of bezuclastinib and 37.5 milligrams of sunitinib daily. RDRX is one of the newest trials for GIST patients. This trial is a phase 1 first-in-human study for patients with metastatic and or unresectable GIST. Eligible patients must have documented progression on imatinib, and the patient must have a mutation in the KIT or any PTGFR mutation other than exon 18. Patients are now eligible if they have previously been enrolled in the NB003, which is the Nube trial, the Theseus THE 630 trial, or the PEAK trial by Cogent. Earlier this summer, IDRX did share early data which showed significant response in KIT exon 13. Mice models that were used in the study were treated daily with imatinib, sunitinib, avapritinib, or the IDRX 42 drug, and data showed that IDRX 42 caused tumor volume shrinkage compared to baseline, as well as tumor growth delay and significant decrease in mitosis. One thing to note is that this trial only has one trial site currently in Pennsylvania at Dana-Farber. On this screen, I have shown 5 trials that are currently recruiting for SDH deficient patients. It is amazing to see how much we have progressed in the GIST world, where we once didn't have any clinical trials for SDH patients, and now there are 5 actively recruiting studies for both USA-based and global. Belzutifan is a trial by Merck Sharp that is a phase 2 trial to evaluate the efficacy and safety of the drug. This trial is open to multiple cancer centers, and this trial is it's not just GIST specific, so there's other cancers as well, which include neuroendocrine tumor, advanced pheochromocytoma, paraganglioma, and also advanced SDH deficient GIST. GIST is evaluated in the study's cohort C, and it is for patients who have locally advanced metastatic or unresectable cancer. This study is international, as I had mentioned, multiple centers, with the most trial sites amongst all the studies I will talk about today. Within the United States, there are trials in California, Iowa, Maryland, Michigan, Missouri, New York, Pennsylvania, Tennessee, and Texas. Internationally, there are sites in Australia, Canada, China, Denmark, France, Germany, Hungary, Israel, Italy, Japan, Netherlands, Russia, Singapore, Spain, Sweden, Turkey, and UK. Like, there are so many trial sites, and it is amazing to see that many for an SDH deficient clinical trial. Next is rogoratinib, which is a trial by the NIH, National Cancer Institute, that is a phase 2 trial studying the efficacy of the drug in sarcoma. The trial has 2 cohorts, each for a different type of sarcoma. So cohort A is for patients with an alteration in FGFR, which is fibroblast growth factor receptor, and cohort B is for patients with advanced SDH deficient GIST. This phase's objective is to estimate the progression-free survival, as well as assessing safety and tolerability. The study is USA only, with trial sites throughout the country. So the trial sites are in California, Colorado, Florida, Illinois, Maryland, Massachusetts, Michigan, Missouri, New York, Ohio, Pennsylvania, Tennessee, and Texas. The next trial is Inhibrx 109, which is a trial by Inhibrx that is a phase 1 open label, and it's a first-in-human study with patients with locally advanced or metastatic or unresectable solid tumors, including sarcomas. Again, this is another trial that is not just GIST specific, but GIST is included. It is in the expansion cohort of this study. The study, like I had mentioned, is not just GIST specific, but there are other solid tumors, including Ewing sarcoma, chondrosarcoma, and colorectal adenocarcinoma. This trial is available similar to regorafenib, which is USA only, but there are multiple trial sites, including Arizona, California, Colorado, Georgia, Illinois, Michigan, New York, Ohio, Oregon, Pennsylvania, Tennessee, Texas, and Virginia. Next, I want to talk about temozolomide, or TMZ for short, which is a trial sponsored by Dr. Adam Burgoyne, who is a GIST specialist at University of California in San Diego. This study is a phase 2 open label trial understanding the efficacy for patients with advanced or metastatic SDH deficient GIST. Treatment for this trial will continue for 6 months with an option to continue treatment after 6 months if a patient is still benefiting. A patient will stop prior to 6 months if there is disease progression or unacceptable toxicity. This study is USA only, with trial sites at UCSD in California, University of Miami in Florida, OHSU in Oregon, and Fox Chase Cancer Center in Pennsylvania. Lastly, I want to talk about NEO-B or NEO-RAY, which is a trial by Novartis that is a phase 1 study in adult patients with advanced or metastatic cancer of any of the following cancers, which include breast cancer, lung. I truly do apologize for that. As I was saying, for the last trial, which is a trial by Novartis, this one includes cancers including breast cancer, lung cancer, prostate cancer, and GIST. This trial is for patients who have no standard therapy, which is available for them, or they can't tolerate a medication. GIST will be studied in cohort C of this study. This is a multi-center global trial similar to belzutifan, with trial sites in the US, including City of Hope and Stanford University in California, Johns Hopkins University in Maryland, OHSU in Oregon, Pittsburgh University in Pennsylvania, and Lifespan Cancer Institute in Rhode Island. I do want to mention that the trial site in Lifespan Cancer Institute in Rhode Island is not yet recruiting, but it is listed to start recruiting soon. Internationally, it is available in Austria, France, Netherlands, Spain, and the UK. On the topic of SDH, I do want to share about the NIH Pediatric and Wild-Type GIST Clinic. This clinic is a collaborative effort between clinicians, research scientists, and advocates who share the goal of helping young patients with GIST. The annual clinic brings together GIST experts from all over to generate a wealth of information to discover the underlying mechanisms behind pediatric and wild-type GIST. The 2023 GIST Clinic will be held September 20th to the 22nd, and they are currently accepting applications for patients who are wild-type to be seen at the clinic. If you have any questions, please reach out to BJ Thomas, who is a nurse navigator at the NIH, or Sarah Rothschild, who will be able to provide any additional details. So this concludes the presentation. I truly hope this was helpful in understanding mutations and getting an update on what is going on in GIST clinical trials. Being an advocate for yourself or loved one is so important, and the best way to advocate is to ensure you stay up to date. We will now transition over to a Q&A portion. Please, if you have not already done so, use the Q&A feature to ask your question so we will be able to discuss it. Thank you. Okay, so one question that we have so far is, I have recently had my first metastatic recurrence for GIST and had surgery a couple of weeks ago to remove it. I'm awaiting my mutational testing report. Does that mean that GIST is not responding to imatinib anymore? What is the likelihood of a second metastatic recurrence in a different mutation? I am still on imatinib until I get back the mutational report. So that is a great question. So it is important to note that a way to know if a secondary mutation has started is either from any type of progression that has occurred, or if a tumor, which was before responding really well to the medication, has now progressed or has become metastatic and is no longer responding to either a higher dosage or a different medication. I would definitely recommend talking to your oncologist, if not your GIST specialist. If you're not seeing one, definitely reach out to us and we'll be able to get you connected with them. But asking them what if they have gone ahead and tested you where they had tested. So let's say you had your primary, I'm reading to see if you had listed your primary tumor. If you had put your primary tumor in your stomach and then you had a recurrence in your liver, there could be a chance if you had recurrence that there was a secondary mutation. But again, it's important to speak to your doctor to see if the medication is no longer responding, what they can do if it is doing ctDNA or obtaining a biopsy to test that as well. My other suggestion would be to wait until you get mutational testing back to see if your medication is a medication that is responding to that mutation. And then I would recommend staying still on imatinib unless your doctor tells you otherwise, and getting the results for your mutation. Another question that we have is, did you say that belzutifan trial is also accepting people with neuroendocrine tumors? Is there a relationship between GIST and neuroendocrine tumors? I've had both of these cancers and did not know of any connection. So trials, especially ones that are not GIST specific, may also be testing multiple other cancers. It does not necessarily mean that there's any type of connection between the two, but it could mean that since they're both sarcomas, that a medication may show response to either or. So that's the importance of clinical trials, is because they will test different cancers to see which ones respond really well. As we know, like for example, for a Gleevec doesn't just show response to patients who have GIST. There's other cancers that imatinib, believe it or not, also is used for those other specific cancers. So that's why clinical trials may have other types of tumors that they also, or other types of cancers that they also test. We have another question regarding belzutifan on do we know the preliminary results for any of these clinical trial phases that you mentioned, including belzutifan? We don't have any data just yet, but we will definitely share as soon as we get more information from these trials and share them with all of our members. We have one question: can hand and foot burning or itching be an indicator of recurrence in a patient with previously resected GIST? No medication treatment, but 6-month MRI shows a lymph node. Definitely get that lymph node checked out or biopsied, or even sometimes what doctors will do is they will continue monitoring it on imaging to see if it changes in terms of size. Hand and foot burning or itching can be an indicator of side effects from any prior treatment that you may have been on and it has just not been resolved. Definitely using interventions like urea cream or a pumice stone to help with some of that. The hand and foot burning or itching, if it is caused by sores, is definitely something to look into. We have one question. I was taken off of Gleevec after 12 years. I had advanced metastatic GIST. Was it right for me to be taken off Gleevec? I had clean scans for 10 years. That is amazing to hear that you've had clean scans for 10 years. This is a question definitely to talk to your oncologist or your GIST specialist with. Definitely having that discussion. And if you feel that you were not having any side effects and you felt more comfortable staying on the Gleevec long-term versus having to come off of it, that is a conversation I encourage you to have with your doctor. There's another question, is there an update on the trial of 5 versus 3 years of adjuvant imaging? At this point, we don't have much information on that, but again, as soon as we get more updates, we will have another clinical trials and other data update where we will be able to happily share information on that. I'm reading another question. What is the current general feeling for CT with contrast versus MRI for monitoring progression? And what is the ideal interval for imaging? Great question. The ideal interval for imaging is definitely specific to different patients, and that's a conversation, of course, to have with your doctor based on what we have learned from our patient registry. Patients, what they start getting concerned with CT scans is the contrast and the dye, the radiation that they continue to get. And what some doctors, what they'll do is they'll start alternating between CT scans and MRIs to decrease some of that contrast. Some doctors have also mentioned that they use MRI exclusively, or some doctors will mention that they do a CT PET scan for one round and then an MRI for another. So it really depends on your oncologist. As long as a patient is getting surveillance and imaging, I think that's the most important part. But for the most part, CT scan is typically the general type of imaging that is done. We have a question. My GIST was diagnosed in November of 2019. It was a large tumor. Exon 11 is my mutation. Which drug is best suited to treat exon 11? That is a great question. As we know, Gleevec or imatinib is the standard recommended first-line treatment for exon 11 patients. It's important to know exactly what amino acid or DNA change within exon 11. And then the next standard treatment for exon 11 would be Sutent, followed by Stivarga, regorafenib. So that is the general way of getting treated for your exon 11. Are you on any medication? If you are, please share with me. We have another question, which is a great one. Do any of the trials administer placebos, or all participants receiving the actual drug being tested? That is a fantastic question. So for the most part, the trials that I have mentioned, if not all, they don't have any placebo. So the important thing to note on those is the term open label. So what open label lets us know is if the patient gets to know which drug that they're taking. Randomized means that if you have a group of patients, they are randomized on who is getting it. There isn't a certain criteria that they have to follow on, like, this person gets this one because we said so. It's randomized, but it is open label, so you do know which drug you are getting. We have another question on any upcoming trials for NF1 or alternative treatment options. At this point, we do not know of any that are NF1 specific, but definitely keeping a lookout for that. We have another question, which is, are there other reasons why tumors become resistant to a drug? And if so, what are they? I have metastatic exon 11 GIST, which has become resistant to Gleevec, Sutent, and now I am on Quinlock, which has been working for a year. But I had blood mutation tests done twice, and no new mutation has been found. That is a great question. It is, for the most part, what we understand, the reason as to why tumors become resistant to a drug is because of the either there has been some sort of secondary mutation that has been caused, or perhaps there's a different change that has happened. I think it's important to take a look and see exactly the differences between the blood mutation tests done, and then if compared it to the actual tumor sample mutational testing done. Because as I had mentioned earlier, ctDNA or blood sample tests they are very effective in patients who have a higher tumor burden or more tumors in their body because it sheds more cells into the bloodstream so that once the bloodstream, and so that once we get the blood, we extract the DNA from it. So it could be a possibility that the ctDNA that is being done is not extracting enough where they're not able to fully see the whole picture, but it's definitely something to go back if you've had metastatic GIST, if they are able to biopsy the METS locations to see if they're able to get a different reading. We have another question. Can we do mutation testing again after 4 years? It is possible. Anyone can get mutational testing done over and over. However, if there isn't any type of indication as to a secondary or trying to figure out something else, I don't think there's any necessarily a reason to get mutational testing done again, especially if it's the same tumor that was resected prior and then had mutational testing done. The only way that they would do mutational testing again is retesting the original tumor sample since there aren't any other metastatic disease that has occurred for them to test. I'm looking to see if there was any other questions. Please, if you have any other questions, please go ahead and share with us. So here we have a question. I am KIT exon 11, developed imatinib resistance, and determined to have a secondary KIT exon 17 mutation. I have been on regorafenib for almost a year now. Would I be eligible for the PEAK trial? I would be concerned about being placed on the control arm sunitinib, since I already know because of my mutation that sunitinib would not be effective. I would suggest you reach out to me privately to go more in depth about this, but in terms of the PEAK trial, the mutation, because it is a KIT exon 11 primary with a secondary KIT exon 17, it would be more so for the perhaps the new INSIGHT trial with Decipher, but at the same time, I do notice that you have been on regorafenib, and since the Decipher one, it is for second line, you wouldn't be eligible, but I'm not entirely sure about the sunitinib for the PEAK trial, since, as you mentioned, for KIT exon 17 with the sunitinib arm, but definitely we can talk about that offline if you would like. We have another question. Does the Raft group patient registry keep data on all side effects and reoccurrence of GIST and subtypes? And does a yearly summary or report be published? Absolutely. I think that's a fantastic question. The Raft group does have their patient registry. We do collect any and all side effects that are reported by patients in our registry. And that includes any reoccurrences that a patient may have, any progression that they may have in GIST, and all the subtypes, so primary mutations, secondary mutations. And we do have a yearly summary or report, which is published. It's called Patient Registry and Review, and you are able to find that on our website. Waiting to see if any other questions. So we have another question. How can someone join the GIST patient registry? I think that's a fantastic question. So in order to join the patient registry, you are able to reach out to us. So the email to reach out to us is patientregistrydepartment@liferaftgroup.org. And in there, you can send us an email letting us know your interest, or you can go on to our website and become a member of the Raft group. And in there, you are able to provide us with some of your basic GIST information, and then one member from the data management and research team will then reach out to you, welcome you to our department, our organization, as well as our registry, and go over a couple of questions and be there for you throughout your entire journey. The benefits of it is that we're able to be there for you and track your GIST journey over time, and then also share any types of updates or clinical trials that you may be eligible, as well as participate in some clinical trials that we are able to share with you, or even some other types of activities that we have. Another question we have is, can you please post your email again? Absolutely. I will go ahead and share it in the chat. Another question that we have is, what % of database participants keep up with updating their data, and how does the quality of life submitted data submitted is evaluated? So that's a great question. So one of the best things about the patient registry, especially our GIST patient registry, is the amount of activity and participation that we have from patients. Even when you look at data compared to the general versus ours, like for example, mutational testing, we have over 60% of our patients in our registry who report a mutation, whereas the general percentage is like 20% or less than that. So because of that, and I do believe that's because of the constant relationships that we have, the resources that we offer, and we really try to engage with not just the patients, but caregivers as well, and be the support system that, you know, all GIST patients should have. And because of that, we notice that patients will interact with us and respond to our inquiries of medical updates or providing us their participation in our GIST patient registry. The quality of the data that is submitted is fantastic. We have an extensive quality control that we have, as well as patients always will share their reports with us, whether that be their pathology report, mutation report, any CT scans that they have. It is very beneficial for us, so that once we do go ahead and have them be part of our patient registry, we can use that reports to also further ask more questions or have more information in our registry. And if we do have any additional questions, we can always ask the patient and be there for them to advocate. So we have a question from the UK. Do we ask our oncologists here about applying to take part in one of the trials you have mentioned, or do we contact you? That's a great question. If there is a specific trial that you are interested in, and there is a trial site in the UK, definitely talk to your oncologist about it. If any other questions, you can always contact me, and I'll happily be able to connect you with a clinical trial specialist or a clinical trials coordinator that may be affiliated with the pharmaceutical company, so that you'll be able to reach out to them as well. We have another question. Is there a limit on how many CT scans should a person take in their lifetime? There is no actual data on that. And again, that's something definitely to talk to your doctor about if that is something of your concern. The thing with CT scans is because of the contrast, not all patients may think that it's a something lifelong to continue. And because of that, they may decrease the frequency that they get it. So they may start at the start of their GIST, getting scans every 3 months, and then gradually extending that frequency to 6 months, 9 months, 12 months, every other year, etc. So again, that's a discussion to have with your doctor. These are great questions. I'm happy to have so many of you all ask these. Definitely, if you have any other ones. So we are at the end of the hour, and I do want to once again thank everyone for being part of this webinar. And I truly hope that it was helpful in understanding more about mutations as well as getting a clinical trials update. We will definitely be sharing and providing more updates as we get more information, specifically for the Decipher one. Definitely, if you are interested about that, please contact my colleague Denise Evans. Her emails is devans@liferaftgroup.org. If you have any questions for myself, I have shared my email. I will also share my colleague Denise's email in the chat. Definitely reach out to us. And I hope everyone has a wonderful rest of their day.